Hello everyone. I'm making this recording to get us ready to call transfer RNA genes because our genome has quite a few of them. So when we call the tRNAs, what we do is we use programs that look at the DNA sequence and then they look and see what is transcribed that could potentially be a tRNA molecule. And it's actually fairly straightforward to do that because tRNA molecules are very conserved. They have a specific sequence that allows them to have a specific shape, and that shape allows them to fit into the P site and the A site into the ribosome. They also have to interact with the messenger RNA. Because at the 3' end, the tRNA carries amino acid, and at the anticodon end, the anticodon has to match with the codon and the messenger RNA. So if there's any way where any of that can go wrong, then you can't make a protein. And if that protein is essential for the viability of the organism won't be viable. So these tRNA prediction programs make use of the conservation, sort of like BLAST, in order to find potential tRNA genes within our genome. So I just have a picture here from my molecular biology book. So if you look at these pink things right here, these are the tRNAs, and you would have learned in Gen Bio or your maybe your cell biology class that we have our large and our small subunit of the ribosome, and clamped between them is the messenger RNA, and we read the codons. And here we have the A site and the P site, so we have two tRNAs together, and these are the codons. So you can see that the codon is base paired with the anticodon, with A base pairing with U and G base pairing with C. So we see two tRNAs here, each carrying an amino acid. We've captured the ribosome in the middle of making an amino acid, making a protein, and adding amino acid 6 to amino acid number 5. So here's amino acid 5, and we're just about to add amino acid 6. So this arrow shows the peptide bond being formed between amino acid 5 and amino acid 6. When that happens, this tRNA will be empty, so it will leave, and then there will be a translocation where the tRNA carrying the growing peptide chain will move over, and then you'll bring in your next tRNA. So here's your next tRNA arriving with amino acid number 7. So you can see that if these tRNAs are going to interact with the ribosome, even though their anticodons are recognizing different codons in the messenger RNA and they're carrying different amino acids, that they have to be able to interact with the ribosome in the same way. So this is a structure of a transfer RNA. It is a single strand, but it folds up into this structure. This isn't three-dimensionally what it looks like. I have a picture of that in a minute. It's just easier to draw this way. So if you look at from the 5' to the 3' end, which each one of these dots representing a base, you can see that we have the 5' end, we have a phosphate. This is the acceptor stem right here, which is shown in black, and we have 1, 2, 3, 4, 5, 6, 7. See, I think 1, 2, 3, 4, 5, 6, 7. That's right. 7 base pairs. That's the dots here mean that they're base paired with one another. So that's the acceptor stem, and you'll have 7 base pairs there. There's a stem loop structure. This is the D arm right here because it has dihydrouridine. This is the T psi C because there's this conserved sequence right here where you have a thymidine, which you wouldn't normally expect to have in a transfer RNA, but is there. And then this psi stands for pseudouridine, and then C is cytosine. The acceptor stem right here has this white base right here. That's a discriminator base. That helps you discriminate between the amino acids. And then there's a CCA, and then you have a hydroxyl group at the 3' end. This picture right here shows conserved positions. Any place where you see an R, that's a purine. Where you see a Y, that's a pyrimidine. And if you see a specific base, that means all of the tRNAs have the base at that position. And here's our anticodon down here. tRNAs have a lot of modified bases. We're used to thinking of A's, C's, U's, and G's, but there are lots of different ways we can modify this. So a pseudouridine, that's where the psi comes in. This is dihydrouridine. So these are uracil derivatives. We can methylate. You can add a lysine. There are lots of different ways to do this. In fact, about 1 base in 10 is chemically modified, and there are more than 60 modifications known. And probably these modifications facilitate codon recognition, or maybe ensure that the amino acid that's attached matches the anticodon. So if we look at the primary structure, again, we're going from 5' to 3'. If we're looking at just one strand right here, that one strand folds up right here to form the acceptor stem and the D and the T size C loop and the anticodon loop. And you can see generally, but not always, you have the GC base pair and the A's and U's base pair. Again, not strictly. But this shows the secondary structure, and you see the tRNAs emphasized that way because it's easier to draw. But I just wanted to let you know that this is what it really looks like. So when you look at the three-dimensional structure, you get this highly folded structure right here where the bases are inside and accessible to solvent. And in this picture right here, that's the amino acid right here attached to the acceptor stem, and down here that is the anticodon. So we will look at the secondary structure like this, but I just want to make sure that you know that the tertiary structure is what it really looks like. So we do maintain that tertiary structure by stacking interactions and base pairing. And again, these bases have to be invariant or semi-variant across all transfer RNAs, no matter which one of the amino acids that they're carrying or what their anticodon structure looks like. And again, that's necessary because all tRNAs have to interact with the ribosome in a similar way, no matter what the amino acid they have or what the anticodon sequence is. So simply that's shown here. So if we're looking at our messenger RNA from 5' to 3', we're looking at a DNA sequence. So our DNA sequence is the same as the RNA sequence, except for the U replaces the T in the DNA. So our codon reads 1, 2, 3 like this. And remember, we have anti-parallel whenever we have two nucleotide strands base pairing. So A pairs with U and U pairs with C, usually, but because we always write in the 5' to 3' direction, we would express this as GAU, even though it looks like that's backwards in the in this picture right here. So if you wanted to write down the anticodon structure for this AUC codon, you'd write it as GAU to write it in the 5' to 3' direction. The other thing to keep in mind is that when the transfer RNA is first transcribed from the DNA, it's in what we call a primary transcript. So it's much longer than it will eventually turn out to be. So there are enzymes called RNases that will do some trimming. So here we're doing an RNase P cut right here and an RNase D cut right here. We have to make sure that we add this CCA. We will do all those base modifications that I talked about, and some of these bases get spliced out. So you can see that the mature tRNA right here is a lot smaller that we end up with, but we can see it's nicely base paired. We have all of our loops. We have our 5' phosphate right here and our CCA right here, and this is an example of a mature tRNA for the amino acid tyrosine. Here's another picture right here. So this doesn't show the processing nearly as maybe as clearly as the last one, but I liked how it showed that you get the amino acyl tRNA at the end, and so when you say amino acyl tRNA, so this is the mature RNA right here. When we say amino acyl tRNA, that means the the tRNA has the amino acid attached. So that's what that little pentagon is supposed to be. It's supposed to be that's the amino acid, even though amino acids don't look like pentagons. So again, 1, 2, 3, 4, 5, 6, 7 base paired at the acceptor stem. 5' phosphate, that's our discriminator base right there, and then CCA right here. So they just wrote N for just random nucleotide. So we have enzymes that are responsible for attaching the amino acids, and they have to be very good at their job. They have to make sure that when you look at this anti-codon loop, that the amino acid that they attach is the right one. Otherwise, you will incorporate the wrong amino acid into your protein. So that discriminator base helps the enzyme make sure that it does the right attachment. So this is from the bioinformatics guide, and again they show the T loop and the anti-codon loop. So here again they show the 1, 2, 3, 4, 5, 6, 7 base pairs at the acceptor stem. That's our discriminator base that helps the enzyme attach the right amino acid to the right anti-codon, and there's our CCA right there. So what we're going to do is we're going to call our tRNAs using two different programs. One is Aragorn and one is tRNA SE. When we run the auto-annotation, which we've already done on our genome, DNA Master has built in an old version of Aragorn. It's version 1.1. There must be some reason why they can't do the old one. I don't know why, but it can't. So it will call some basic tRNA genes, but it won't call them all, and it might not trim them. So what we need to do is we need to make sure we use a later version. So what I'm going to do is, this is a link to the newest version of Aragorn, and what I'm going to do here is I'm actually going to run this because it's very fast. So it says I can upload a FASTA file. So there's my Frankenweenie FASTA file. So if I just hit open, and I'm going to call both tRNAs and tmRNAs. I don't think our phage has any, but I'll just call both anyway. We won't allow introns. This is linear, both strands, and standard output. So if we do that and just hit submit, it's pretty quick. The other thing that we should note is this is Aragorn version. I'm going to write this down, 1.2.41. So we are using a later version. We're going to have to note that in our notes, and we will reference this paper when we write our genome announcement. So what it does is it gives you a whole list of all of the tRNAs. It draws little pictures of it for you, and some of these might not be real. So if you look at this, I don't think this is a real. This doesn't look like a real tRNA to me. Or if it has this really long loop, you know, again, well, maybe that's real. I'm not sure. So that's why we use a number of different programs. So what it does is it gives you the coordinates of the tRNA, but it's up to us to figure out what's real. It will also tell us the amino acid using the 3-letter code. So this is isoleucine, and it will give us the sequence of the anti-codon. All right, I'm going to go back to my PowerPoint. We can use another program called tRNA Scan SE, and that is better at identifying tRNAs that don't follow the strict guidelines, but the tRNAs might have a slightly enlarged loop or shortened stem or be missing one of those conserved bases that I showed you in those other pictures. Maybe Aragorn doesn't identify them because of that, but if we relax the parameters, then tRNA Scan SE might identify them. But it won't trim the ends properly. So that's why we have to use both. Okay, so I'm going to go to, let's see, that's our bioinformatics guide right there. So here's the tRNA scan. We are going to use version 2.0. We should make a note of that too. And when we run this, so we have to set this at bacterial and the default options and run. Okay, so bacterial default, upload the sequence. Score cutoff is 0, and then you click the Run tRNA Scan SE button. Okay, so let us do that. Let's go back to my PowerPoint and go to tRNA Scan SE, and okay, not finding a scroll bar for some reason. Okay, here we go. All right, so I'm going to change this to bacterial, and run the default. And oh, here's my scroll bar, sorry. It's just off the screen right here. I'm going to do the same thing where I'm just going to put in the FASTA for Frankenweenie. And do that. And score cutoff is 0. So we're going to go ahead and run the scan. I think that worked. Okay, so I did do this earlier. There we go. Okay, so this one takes a little bit longer, and again, it's a little shorter if you don't have as many tRNAs or if your genome is not quite as long. But it looks like actually it is finished. So tRNA Scan gives you this nice table, and it tells you the beginning and the end of where these tRNAs are, and tells you the anticodon and the amino acid. So here it says that Frankenweenie has potentially 16 tRNAs. If you go here and hit view, it'll actually do a nice little drawing for you, and here you can see, well, does it really look like a tRNA? And this does. So there's our 1, 2, 3, 4, 5, 6, 7, and then there's the discriminator base. The CCA isn't there, but that can be added later. Okay. All right, so what I want to do now, I think, is go back to DNA Master. And if I go to DNA Master, I want to see what tRNAs that the old version of Aragorn called, because those will be there. And you can, if you look right here, if you see where it says all, you could just look for tRNA, and there they are. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay, so we're going to have to reconcile these programs, what we're seeing right here. tRNAs are usually transcribed sort of in little islands, and you can see that we have some here around 94 or 95,000, and we have a whole bunch around 133, maybe to 136,000. So the next thing we can do is we can go back to Famerator, and if we go to Famerator, the tRNAs aren't shown on our drafts, but I'm going to look at just because they will be shown here because that's a published genome. So if I look, again, this is going to be tricky, and partly this is tricky because we only have 6 of these. So what am I looking for? I'm looking for the first ones, 94 to 95,000. So this is a little tricky because usually you would expect to see the tRNAs and you wouldn't expect them to overlap with protein genes. So I'm going to look and see what happens with just because, and I want to find the same version in the genome. So here, this is FAM7825. Let's follow the purple, and it's right here. And I can see right here that I do have some tRNA genes called here, and then they have one tRNA called here. So, but you do notice that in just because the tRNAs don't overlap with the protein coding genes, and they don't. So that's something that we're going to have to look for when we look for where Aragorn and tRNA Scan, as he calls them. So that's maybe a source of, let's see, just because it has 4, ours had 3. So again, we'll have to look at those. Okay, where's the other one? The other island was sort of like in the 133 to 136,000, and we can see there's a lot of space here. So again, let's look at a similar spot in just because. So that's 5979. Where is that? Well, maybe we don't have that one, or maybe 9. Oh no, that has 9 family members. We should have that. 5979. Not seeing it. Okay, how about if I look at, oh, I don't know. How about 7455? See if I can find that. Yes, it's right here. Okay, so I just want to make sure I'm looking in the right spot. And oh, and there's that little gene right there. 10,707, which is here. Okay, so it looks like, if I can get this on the same screen, that in just because there are 3 tRNAs before that little gene and a bunch after, and one kind of squeezed here after this FAM7535. So we might have the same thing where we might have some tRNAs here, some tRNAs here, and maybe we're going to squeeze a tRNA there. So it does look likely that we have tRNAs in Frankenweenie in the same place that we have them in just because. Okay, so I told you about trimming. So what we need to do when we call this is we have to make sure that we put in the right 5' and 3' ends. So this is from the CV Phages guide. So again, we've got our 1, 2, 3, 4, 5, 6, 7. There's our discriminator base, and then we have CCA. So the way that this works is that when we identify this by, so here's, this is Aragorn version 1.2.38. We have 1.2.41, so ours is even more up to date than that. So it has to be at least that up to date or more so, and we have that. And our tRNA scan is version 2.0. Okay, so we've done both of those. And then we have to say, well, is this a tRNA? And I think, again, we're in unusual territory here with our phage. Usually with most phages, they don't have many tRNAs. Some don't have any, some might have 0 to 1s, but some might have 30. Looks like we might have, I don't know, 15 or 16. So I think we found those tRNA clusters. So it would be unusual if it has coding potential. So we have to look at our coding potential match. And as the opposite strand is a number of other phage tRNAs found in the same genome, or at a distinct location from other tRNAs. So we did find these clusters. It says it doesn't mean that we are wrong, but anyway, that's what we're looking at usually. So here, the best tRNAs have the tRNA SE infernal score higher than 35. Okay, so let's go back to tRNA, the tRNA scan SE, and there's our infernal score right there. So it looks like we have everything but this last one looks like it's above 35. So the infernal score is something that we're going to want to put in our notes. All right, let's see. I already forgot where I was. Okay, oh, that's right, we were trimming. Sorry about that. Let's go back to the, okay, we're back to the C phages guide. The tRNA shouldn't be more than 90 base pairs. It shouldn't be within a protein coding gene. It shouldn't be in the middle of but the opposite strand of a cluster, although we have found examples that are real. So here's the trimming. If the 3' end of the sequence is not CCA, it should be trimmed at the first deviation and the remainder shouldn't be called. Okay, so if I go back to this tRNA right here, okay, so this is, so here's our 7 base pairs, that's our discriminator base, and that can be any one of the 4, and there's no CCA. So this looks to me like it's been trimmed properly. So that is the first one, and if I go back to tRNA scan SE, and that's the first one, so that begins at 133,509 to 133,583. That encodes a tryptophan. The anticodon is CCA, and the infernal score is 69.5. This is tRNA scan SE, and it's version 2.0. I'm going to go back to see what Aragorn does with that. If I go back to Aragorn, remember I saw some things on Aragorn that I wasn't sure that were real. So let's look and see if we can find this. That was what, 133,000, 509, 1335. Now this says 133584. Yep, okay, tRNA scan did not call this properly. This is called properly. Okay, so we would refer to that as 133509 to 133584. And the reason for that is that we've got 1, 2, 3, 4, 5, 6, 7. There's our discriminator base, and it goes CCA. That's a C, so we can keep that C. Okay, so if we go back then to DNA master, and if I can find this 133509 is right here. See, this is 133509 to 133586. We're going to have to change this to 58. Actually, here, let me show you before I get here. If I go to where it says product, all right, okay, see where there's a C right there, and then it says UG? Remember, that's supposed to be CCA, so we have to get rid of that U and that G. Okay, so this is going to be 133584. And then I'm going to hit the calculator, so it's 76. And now I'm going to hit post and then validate, just to be sure that I have everything properly. Okay, so I'm going to go back here to my description. So I've trimmed this properly. What I have over here, this is tRNA, and I wanted to put this here so I can give an example of proper notes. So this is trimmed properly. The length is 76, it's not greater than 90. This says tRNA, this is tryptophan, CCA, is what Aragorn said, and that's right. So in my notes, I'm going to say that, let's see, I'll go right here. Trying to get both of them. Okay, so this is what we did for tRNA. So we said tRNA, GLY, TCC detected by both Aragorn and tRNA Scan SE, and we had the infernal score. Ends have been trimmed to match Aragorn 1.241. I'm actually just going to copy that because this was in fact accepted. So I'm just going to modify this. So this is tRNA trip. TRP, and this was, they want that lowercase here. I don't really like it, so I'm just going to do CCA in my notes. Detected by both Aragorn and tRNA Scan SE. I'm going to add here that this is a version 2.0. Version 2.0, and the infernal score was 69.5. And then I say, ends have been trimmed to match Aragorn 1.2.41. And now I'm going to hit post. So this is an acceptable way to annotate a tRNA. So what we have to do now is we need to go through all the rest of them and use the same kind of strategy and do the same thing for the rest of them. So I hope that helps, and I will see you at our next meeting.